Andreas, the floor is yours.
Great. Thank you very much. Can you see my slides?
Yes.
Good. So I would like to start out as well to thank the IFCA, and I think not only as being supported, but as been pointed out before, I think it's really unique to have a foundation which is so targeting a relatively small disorder, congenital disorder, and it really brings together researchers, clinicians, and patients, and I think it's a fantastic initiative. Now, before I start, so my group is actually more developmental biology group, and we are interested in how organs are forming, what are the molecular programs that are driving this formation, and then also how an organ is maintained during adult life. And of course, one of the organs we are particularly interested in is the adrenal gland. Rather than going into too many details about the experiments which we are doing, I would like to use that talk as a platform to discuss a little bit stem cell biology in the adrenal gland. And I think it's very important, of course, if we want to use stem cells for treatment. And as you, don't know whether this works. Yes. So as you know, the adrenal gland is a zonated organ. So we have the zona glomerulosa, zona fasciculata, in humans as well, zona reticularis, which produce the different hormones. And then the medulla, which is developmentally from a different origin. So it's neurocrest derived. And all of this organ is surrounded by the capsule. Now, it has been recognized many years ago and really shown by David Broad the first time that the maintenance of the organ is through a kind of centripetal mechanism. So what that means is that we have cell death at the, which is occurring. Oops, doesn't work already. Can I go back? How can I go back? Right. Yeah. So cell death, which is occurring at the boundary to the medulla, and then the cells are actually moving down, and in this movement, there's cells coming from zona glomerulosa, which move further away from the capsule, and this leads to a transdifferentiation. So there's this centripetal cell migration. Proliferation is occurring mostly in the outer layer, so in the zona glomerulosa and the outer zona fasciculata. And then cell death is occurring a bit further down. So to study and to understand the turnover, it is, which I think is very important if we are thinking about using cell replacement strategies. We make use of cell lineage tracing analysis, and Leonardo has already mentioned that. So what does that mean? It means essentially we are labeling one particular cell type using a fluorescent protein. So here, green fluorescent protein. And this is a permanent label. So the cells, when they are proliferating, when they are differentiating, they are maintaining their label. And this allows us then to follow over time what is happening to those cells. And particularly this is important or interesting when we are looking at stem cells, because we can label stem cells and then see what is happening to them over time. And we use that strategy by labeling cells of the zona glomerulosa. So here, these are these rosette type structures, which are labeled here in green. And if we use that Cray strategy, we can see initially, as expected, only these zona glomerulosa cells are actually GFP positive. And then over time, we see that gradual labeling, which very rapidly replaces the entire adrenal gland. And this essentially tells you that in the mouse, we have a very rapid turnover, which at least for me was very surprising. So in females, every 3 months, we have a replacement of the entire adrenal cortex. And the second thing which was surprising is that in males, this tissue turnover seems to be much slower. In fact, it seems to take almost 3 times as long. And this is even though in mice we have this sexual dimorphism, that the mouse adrenal cortex, male adrenal cortex is actually thinner than in females. Then we use the same strategy to target a second progenitor cell population or stem cell population, which Leonardo has already mentioned. So this is actually within the capsule. So these are the cells which during development give rise to the adult adrenal cortex. And here you can see on the right-hand side that again we label in green just the capsule with this approach. And over time, then again, we see cells moving in. What is surprising here is that, again, in females, we see that there's this recruitment occurring from the capsule, whereas in adult males, there seem to be very little occurring. So this fits actually with proliferation studies, which we have done, showing that there's much higher proliferation occurring in female adrenal cortex compared to the males, and that this recruitment of capsular cells occurs to be seems to occur predominantly in the female cortex. So this is just a summary of what I have just told you. So we have essentially two main progenitor population or stem cell population. Really the major one under homeostatic conditions, under normal conditions, are cells which are located in these rosette structures of the zona glomerulosa. They are becoming activated, move down or move towards the center, lose their contact with the capsule, and then become zona fasciculata cells. And then in females, and to some extent also in males, especially under stress, we have these capsular cells, which are also being recruited and then change their characteristic from more mesenchymal phenotype to these steroidogenic cells. And what we have shown, I didn't show you the data, that this, both the recruitment as well as the proliferation, is under control of the gonads, in particular testosterone, so it's actually suppressed by androgens. And for example, if we are treating female mice with testosterone, you can actually suppress the recruitment of these cells. So these are the two major components as far as we know. Then some years ago, we have shown that there's yet another progenitor population, which is actually very similar to the adrenal gonadal primordium, so a very early progenitor population. And these cells, in particular in adult mice or under hormonal stress, can also be recruited and form steroidogenic cells. And then in addition, Stefan Bornstein's lab, they have identified a few years ago a nestin positive population. So this is a developmentally, again, a very different lineage. And these cells, in particular under stress, seem to be able to also differentiate into the adrenal cortex or adrenal cortical cells. I didn't integrate in here the DLK1 positive cells from Leonardo, mostly really because I'm not quite sure how to position them. Are they similar to the GLI1 positive cells? And perhaps we can discuss that later on. Okay, so here we have 4 different populations. Now we could use, of course, those for these stem cells to use what I call the adult stem cell replacement therapy. So here the idea would be to isolate the cells, perhaps with a marker like DLK1 or nestin, put them in culture, then correct them, expand these cells, and transplant them back into the patient. Now, in most cases, the difficulty is that there is no good molecular markers for isolation. We have heard that DLK1 might be one. We need to have access to sufficient cells in patients, and the culture and expansion of these cells, at least in our hands, has been very difficult. The alternative approach would be use what I call a developmental approach, where we are isolating cells from the patient, reprogram them towards the iPS cells, and we know how to do that, and then use those iPS, so these induced pluripotent stem cells, to perform gene correction. And their CRISPR-Cas9 approaches have come along quite well, so we can also do that as well. And then here the idea is to differentiate those cells into adrenocortical cells, or adrenocortical stem cells, I should say, and transplant them back into the patient. So one approach to do that, the approach we are taking is to study development first, to try to get inspired by development, because after all, we would like to use cells which are as similar as possible to the normal developmental pathway. And to do that, it's important to have a look at how the adrenal gland is forming. And here on the left lower side, you see a scanning electron micrograph, which is a cross-section through a developing embryo, and what we call in yellow the lateral plate mesoderm and the intermediate mesoderm. So these are two structures which are developmental structures, and it's really at this interface where the adrenal gonadal primordium is forming. So adrenal gonadal primordium, of course, because this is a progenitor population which will give rise on the one hand to the adrenal progenitors, which are SF1 positive, and then to the gonadal primordium, which are SF1 and GATA4 positive. So we are moving towards that, but of course it is not as simple as it looks on the paper, because we need to have the right initiating factors which are driving those cells into the lineage which we want them to go. And to get some inspiration, we teamed up with the lab of Serge Neff in Geneva, who is an expert in single-cell transcriptomic analysis. And what they have been doing is they mapped at these very early stages of adrenal and gonadal development the expression profile of thousands of cells over time. So you see here an embryonic stage, 9, 10.5, 11.5, and 12.5, and there's many, many different cells which have been on a single-cell basis analyzed. And then looking at marker genes, we can identify different regions in there. And here on the top, I put a circle around that. These are the adrenocortical cells, where you have already actually a differentiation into either the zona glomerulosa type cells and the zona fasciculata cells. So using these data, we can then perform trajectory analysis to see how those cells are differentiating over time. We can hopefully identify further stage-specific markers, which will help us to characterize, develop protocols and characterize better those cells, and then identify signaling pathways involved in the specification. Well, we are not there yet, but we are trying to do that mostly with mouse, but also with human cells to some extent. So we know we are quite well, we have established a very good protocol for the lateral plate intermediate mesoderm, where the right markers are being upregulated. And apparently, in some cases, we now also get colonies which are really reminiscent of an AGP, which is separating, because you can see here on the bottom a colony where you have strong SF1 expression, which don't express GATA4 anymore. So this is reminiscent of what you see in the adrenal primordium. So obviously, we are working towards that with the support from IFCA to improve differentiation protocols, to transplant those cells then under the adrenal capsule, look for long-term engraftment, because that's obviously the aim, that we can have stem cells which are there producing over a long period of times new cells, and then to transfer those to human iPS cells. And I think I am already at the end of my talk. I would like to thank the people involved in that research, in particular the people in my lab, also our collaborators, and of course, very importantly, IFCA for their generous funding. Thank you very much, and I'm happy to take questions.
Thank you, Andreas, for this very impressive talk. Would you see in the long run combination of these gene correction approaches with some form also of transplantation technologies using form of devices? How would you see how this could fit together as one strategy?
Well, I think this fits in particular for these endocrine organs perfectly well. Because obviously, if we are so I think that what was important, at least what I think is important, that we now know that differentiated cells, at least the standard adrenal cortical cells, have a relatively short half-life. So that means that we need to have a system where they are constantly replaced. Now, how you achieve that is one thing. Then when we are using those type of cells in a device, we could imagine that well, they would work in an isolated system, because after all, a lot of this stem cell recruitment is actually driven by hormonal control or control of the body. So it actually responds to signals from the pituitary, etc. And I think that we should have the same system actually if we are having it in a device. So yeah, perfectly, that would fit perfectly well.
There's one question from the chat that I may read to you. Do you think it could be possible to isolate adrenogonadal precursor cell from parts, from the adrenal rest tumors, so to speak?
Well, I think, yeah, I mean, the question is to what extent are these cells identical to an AGP, and to what extent do they maintain their capacity to efficiently differentiate into adrenal cells and to actually, become real adrenal progenitor cells. It's an interesting idea. I haven't actually followed that up at all, but why not? Then I don't know how easy it is to culture them, to expand them. So this would be something which might be worth studying.
Okay, can I ask a question? Yeah, I just wanted to know, I mean, listening to these talks, it's becoming apparent that I guess the field will have to make a choice towards which cell type is best to move forward. And I'm just wondering what you are, Andreas, Leo, and Stefan, what you are thinking about what might be the best cell type. Do you think we should keep hoping in somatic cells? Do you think we should be moving to ES cells or to IPS cells? What's your idea of what may be the next best move? I guess, Andreas, you're for the IPS cells, seeing your talk, but I don't know. I mean, I want to have you guys' opinion on all of that.
Well, actually, you will be surprised. I'm not absolutely for the iPS cells. I just, we have attempted to culture cells. I personally think that as if you can have cells, transplant cells, which are as similar as possible to the endogenous stem cell, this is the best approach. That's why I was always a bit, well, skeptical. I wouldn't say, this is too strong a word, but there's always this caveat when you are forcing the expression that you are still, you are having a very artificial system to some extent, which doesn't mean that's maybe already better than what we have at the moment. So what is better, the adrenal stem cells, or is it the developmental approach where we are using iPS cells and reprogram them? I think this is a question too early to answer, and I would definitely push every funder to support both of these approaches, because you don't know which one will work better. I mean, the advantage of the IPS, as I said, is that we can actually expand those cells dramatically. It's easy to manipulate those, or relatively easy to manipulate them in culture and then differentiate them. As I said, in our hands, we have been unable to culture for a long period of times those stem cells isolated from adult, I should say, mouse adrenals. But perhaps we just didn't have the right medium and the right growth factors which they need to actually start proliferating. Leonardo, what do you think?
No, I totally agree. I think, like, in terms of lineage conversion, is probably the least that might be successful in providing a long-term curative option. And in terms of ES, IPS cells, yes, whichever is able to give us a cell population which is self-renewing, it's good for me. And it's the same major problems that we're having with these cell-based therapies for type 1 diabetes, because even the islet of Langerhans, they undergo a very, very slow self-renewing process, but it's there. So even if you're using these ES-derived cells, if they do not have the ability to self-renew themselves, this is a treatment that might work for a month or for a year, but might not work for long-term. You would need like continuous kind of transplantation or injection of these cells. And Also, if I may add, considering how the adrenal cortex, in terms of the structure, the vascularization, the components of the extracellular matrix, which are quite not random, they're quite, you have mostly fibronectin in the capsule, you have more laminin elsewhere. So does it that one might be an important factor in kind of assessing, in helping cells to, in their kind of differentiation process during self-renewal. And also Stefan has demonstrated through the years a kind of a quite important functional interaction between the cortex and the medulla. So do we need cortical cells only, or there is some sort of crosstalk between catecholamines in kind of helping steroidogenesis directly or indirectly, and therefore we will need a sort of hybrid mini organ also comprising a little bit of chromatin cells. So this, it's a project. Each question is a 3-4 years project.
And I think we should also add that, of course, stem cell approaches eventually will work, and we are looking forward to the new results coming with the ViaCyte. Again, last trials haven't been so successful. In the years, we had a lot of experience now with xenotransplantation, and we have a large xenotransplantation consortium, and I think it's doable. We've been moving a bit now. There's a lot of experience on adrenals, on bovine adrenal cells, very little on porcine adrenal cells. But since we have an established protocol for islet, for porcine islets, for xenotransplantation protocol in humans now, I think it would make sense also to use porcine adrenal cells, and we are characterizing them for the devices. The advantage is obviously that those cells, you don't have to do anything. They are already fully functional adrenal cells, and they may be maintained in these immunoprotective devices. But of course, it's not easy to produce, again, a device that has this long-term efficiency that we would like to see, to be honest.
Yeah, because what's your expectation about having to replace the device? I mean, what's the experience? Do you need to replace it every month, every year? Is it clear what you might expect from that kind of equipment?
So in our trials, we had about maximum a year where there was still some efficiency, and you could think about any also devices that would be more easily replaceable, or parts of it that you could just exchange. And I think this could be a combination for the future, definitely. I think the big advantage, talking about adrenal versus islets, it should be, at least that what we would expect, even easier with adrenals, because we don't have this tight interplay of glucose and insulin, the censoring that is very delicate. But of course, our own energy and the energy of most groups around the world into these approaches for islets have been a lot more extensive than for the adrenals, and we just need to work together now in an adrenal consortium, like supported by IFCA, to just make this happening for the adrenal, what is already on its way for islets. So I think then we should come to the clinic even less than 10 years to be a bit more optimistic. That's also because I'm getting older,
Okay, thank you very much. I think we should close the scientific session and the scientific session of the symposium. And I want to thank you, 4 of you, for this really great discussion and bringing together gene therapy, cell therapy devices to go to the clinics. That's what we discussed at the end, and I think there is a good opportunity there. So thanks for your talks, thanks for the discussion.